let me introduce Laetitia Segin. So she will be speaking instead of Chloé Ferral, I believe, today. She is a researcher at the Institute in Nice, and works in the group of Chloé Ferral, and will have a talk today titled Exploiting Druggable Vulnerabilities for KRAS-driven Lung Cancer. Laetitia, please take it away.
So hope you can hear me, and I can't because someone else is sharing the screen. So, okay, here it is. So do you see my screen?
Yeah, we see the full.
You see the full screen.
The full
like here and like here. Okay, should be good. So I am Laetitia Segin. I work in Ferral Lab, in fact, and I really would like to thank the Anchorage and the joint meeting to give us the opportunity to speak about our job about druggable vulnerabilities for KRAS-driven lung cancer. So I will be really quick on that because everybody knows that, in fact, KRAS is expressed in around, is mutated in 25% to 30% of adenocarcinoma, and that there is no effective targeted therapy yet. So sorry, I will remove my this because I can hear me and it's weird. So targeted therapy has still to be developed. So there is no effective therapy, but in fact, in 2009, there is a new concept that has emerged, the concept of KRAS-dependency. And this concept was really interesting because it did show that even if cells express oncogenic KRAS, they do not need it to survive. In fact, some do and some don't. And it was really interesting. And back then, in 2010, we asked the question why. Why some cells need oncogenic KRAS to survive and others don't? And we hypothesized that maybe the localization of KRAS was important in that concept. And what we found is that, in fact, KRAS localization was totally different in KRAS-dependent cells and in KRAS-independent cells. In fact, you can see here that in KRAS-independent cells, KRAS is localized homogeneously in the cytoplasm and the membrane, but in KRAS-dependent cells, you can see that KRAS form nanoclusters here and here. So then we ask the question why? Why those nanoclusters? Maybe partners matter. So what we found, so a very long story short, what we found in fact is that KRAS was able to interact with Integrin B3 and galectin-3 to form a mega complex that were able to stabilize KRAS at the plasma membrane. This complex was important for KRAS dependency by driving KRAS-dependent cell survival, aggressiveness through the macropinocytosis event and the TBK1 pathway. And what we found, it was that disturbing this complex, for example, with a galectin-3 inhibitor here, we were able to disrupt totally KRAS localization, and we were able to kill KRAS-dependent cells and to inhibit also macropinocytosis. However, one of the issues was that the galectin-3 inhibitor, in fact, with the galectin-3 inhibitor, the issue that we had a lot of compensatory effect with other galectin, like galectin-9, for example. But we were still convinced that targeting this complex was important. So we decided to dig into this complex and to find other partners. And what we found here in Ferral's lab is that CD98HC, in fact, is also a part of the complex. You can see here that when you pull down KRAS in beta-3 positive cells, you pull down also CD98HC. So CD98HC is a part of the complex. CD98HC is an interesting protein because it acts as an integrin modulator. By interacting with integrin, it can modulate its function, and also it's a scaffold for the light chain SLC7 family, like XCT and LAT1. So it's also involved in amino acid transport. So we decided to investigate the role of CD98-HC in KRAS-driven lung cancer. So first of all, we found that CD98-HC is highly expressed in adenocarcinoma and very heterogeneously expressed. And then when we knock out CD98-HC in a very well-known model, the KRAS-driven lung cancer model, when we knock out CD98-HC in that model, we found that we dramatically decreased the tumor burden and also the number of lesions. In fact, we decreased the number of adenocarcinoma, but not hyperplasia or adenoma. So those results, so because CD98-HC is involved in cell proliferation, we checked, in fact, cell proliferation in our tumors, in the CD98 wild-type tumors and knockout tumors. And what we found is that CD98-HC knockout decrease cell proliferation. So this is logical. And in vitro, when we knock out CD98-HC, we decrease the number of colonies. So this it's good. So we are really convinced that this is a good target. But what about patient samples? So in collaboration with Paul Hoffman, we were able to get fresh patient samples from the Tumorotech. So multiple patients, and I will show you the results only in 3 patients, and all of them express CD98-HC. Not 100% of the cancer cells do have CD98-HC, but all of them do have some. So we were able to get 3 patients to sort the cells following CD98-HC expression, low and high expressing cells, and we performed clonogenic assay. And what we found is that compared to unsorted cells, cells that do not express CD98-HC in gray and cells that do have CD98-HC do not form colony numbers. Cells that do have CD98-HC form a lot of colonies compared to cells that don't or to unsorted cells. And when we knock down CD98-HC in the cells that do have it, the red one here, we decrease the number of colonies. So we are really convinced that CD98 is a good target. In fact, we wanted then to see whether if we use an anti-CD98-HC, were we able to disrupt the complex? And the answer is yes. So we used an anti-CD98-HC antibody, a blocking antibody, and you can see that when you pull down KRAS, you decrease the interaction with Integrin B3. So now, in collaboration with Sanofi, what we are able to do is we are developing new drug anti-CD98-HC because the problem of the blocking antibody is that is not really effective. So, oops, I cannot. Okay. So now, because p53 is often mutated in cancer, we wanted to see whether in a KRAS mutated p53 silenced model, we have the same results. And the answer is no. In fact, when we knock out CD98-HC in the KRAS mutated p53 mutated lung cancer model, instead of decreasing the number of tumors and the tumor burden, we've seen the opposite. We dramatically increased tumor burden as well as the number of adenocarcinoma. So we were a little bit surprised. And the first hypothesis at this time was maybe we are able to bypass the effect of cell proliferation, of CD98-HC on cell proliferation. And maybe cancer cell proliferates with or without CD98-HC. But when we checked pCNA, that is a marker for cell proliferation, you can see here that in fact in the CD98-HC tumors, you have less pCNA staining, so they do not proliferate more than the CD98 wild-type cells, cancer cells. So we did not really understand at this time, but we decided to check. So we isolated cancer cells from those mice. So 2 different cancer cells from the CD98-HC wild-type animals and 2 from the CD98-HC knockout animals. And we performed colony-forming assay. And what you can see here is that when you knock out CD98-HC, you totally decrease the number of colony. In fact, they are not able to form colony anymore. As well as in MEFs, when you knock out CD98HC in MEF, they are not able, they form much less colonies. So this means that it's not a problem of the cancer cell by itself, but maybe more the microenvironment. So at this time, so why? So at this time, we were able to, we can see three possible hypotheses. The first one is that adenoma can switch to squamous cell carcinoma. The other one is the origin of the tumor, and the last one is the microenvironment. So we decided to check one by one. So to see whether we have an adenocarcinoma to squamous cell carcinoma transdifferenciation. So this is possible. So it has been shown, in fact, a few years ago that adenocarcinoma can switch to squamous cell carcinoma when you have an elevated amount of RAS, or when you have a matrix softening. And because CD98HC is involved in RAS detoxification as well as in matrix stiffness, we thought it might be possible. Two markers of squamous cell carcinoma have been used in papers to differentiate between adenocarcinoma and squamous cell carcinoma, P53 and keratin 5. So we checked and we didn't find any difference. So all of the tumors are keratin 5 negative, and we didn't see any difference in P63 staining, meaning that yes, some tumors have it, some don't, but there is no more tumors with P63 staining here than in knockout animals than in wild type. So it's not a transdifferentiation. So to understand a little bit better at this time, we decided to see whether CD98HC was an early event or a late event. So we instillated the mice with the adenocre, and 6 weeks after, we sacrificed the mice and we checked for the tumor burden. And you can see here that when you knock out CD98HC, you dramatically increase the tumor burden. So it means that CD98HC knockout, in fact, it's an early event in tumors. So to know whether we have the same aggressiveness in the stage, we decided to check two different markers, TTF1 that is involved in initiation and HMGA2 that is more involved in progression. And you can see that in Y-type tumors, TTF1 is expressed similarly in Y-type and in knockout tumors. However, HMGA2 is much more expressed in CD98HC Y-type tumors, meaning that in fact, we have maybe more tumors, but they are less aggressive. So we did not really understand. However, there is a possibility that it has been shown that that lung cancer, adenocarcinoma, can arise from two different types of cells, progenitor cells from the lung. The alveolar type 2 cells that are in the alveol, and the bronchoalveolar stem cells that are the junction between the bronchiole and the alveol. And in fact, it has been shown that the alveolar type 2 cells are more HMGA2, and the BAS cells are more TTF1. So we decided to check the expression of CD98 by cytometry. And what we found is that CD98HC is expressed mostly in BAS cells and not in alveolar type 2 cells. So maybe there is a difference between BAS cells and alveolar type 2 cells, and the tumor cells that are impacted by CD98HC knockout are only the BAS cells. So this is the hypothesis. The hypothesis is maybe you have a switch from BAS cells origin to alveolar type 2 cells origin when you knock out CD98HC. And what we found, in fact, so we analyzed the origin of the early tumors and late tumors, and what we found is that while we have, so in the KRAS P53 model, when we have around 50% of the alveolar origin and the bronchoalveolar origin, when we have CD98 expression, when we knock out CD98HC, the origin is mostly alveolar. So it's possible that we have a switch between BAS cells and alveolar type 2 cells. However, we will need to investigate further this hypothesis with a specific adenocre that will target specifically best cells or alveolar type 2 cells. However, we know that these cells, the origin may impact, so the best cells or the alveolar type 2 cells may impact immune infiltration within the tumor. So we decided to check whether, and particularly, it has been shown that the alveolar type 2 cells can express the MHC2 and EpCAM. So we checked in our mice whether it was the case. In fact, what we found is that when we knock out CD98HC, we express much more on the epithelial cells MHC class 2 than when we have CD98HC wild type. And however, we haven't seen any difference in PD-L1 expression. And this was interesting because it totally matched the TCGA analysis that we performed with the LUAD system. So we analyzed the CD98HC low and high tumor expressed. And what we found, in fact, it's an enrichment of the MHC2 signature, as well as PD-1 signature and interferon gamma signature. You can see here that we have a totally inverse correlation between the CD98HC expression and MHC class 2, or CITA2, that is the promoter of the HLA-DR. But with the PD-L1, it doesn't work the same. There is no correlation. So we are very happy to find that we are matching the patient samples. So we decided to investigate whether CD98HC expression was able to modulate the immune infiltration system. And what we found, in fact, is that CD98, so you can see here that there is no difference in CD45 positive cells in tumors that do or not express CD98HC. There is no difference in lymphocytes in CD3 positive cells. There is no difference in CD4 and also in CD8. I don't show it here, but we also checked for CD8, and there is no difference in CD8 neither. So for the CD4 plus cells, however, we have a difference when we checked for the CD4 cytotoxic lymphocyte. You can see that when we knock out CD98HC, we increase the infiltration of those cells. In the meantime, we decrease the infiltration of the myeloid-derived suppressor cells. So here we are, and we still need to understand and to investigate how and why knocking out CD98HC increase the number and the size of the tumors in the KRAS P53 model. This is very important because we are developing a drug that will target CD98HC, and we will need to know which kind of patient will benefit from this therapy. Are they only the KRAS mutated one, or also the KRAS P53 one? But also the analysis of the microenvironment lead to the investigation, in fact, of the PD-L1, anti-PD-L1 as a combo with anti-CD98HC, because the MHC class 2 and the interferon gamma could be a good signature for the anti-PD-L1 therapy. And we would like to investigate whether CD98HC could be a good marker for the anti-PD-1, PD-L1 resistance. So thank you for your attention, and if you have any questions. So I would like also to thank, of course, my team and my boss, Chloe Ferral, that couldn't present today, the core facilities and the collaborators, as well as all the funding that help us to develop this work. Thank you very much.
Thank you very much, Laetitia, for a phenomenal talk. There are a couple of questions. I would ask if you could have questions, put them please in the Q&A box rather than the chat. It's easier to see, but I'll try to synthesize. So the first question from Paul Hoffman: how would you integrate other potentially associated mutations such as STK11, KEAP1, or others in these KRAS mutation models?
Yes, this is a very good question, because in fact, KEAP1 and LKB1 change ROS detoxification and will change the environment. So we will need, because CD98HC is a partner of XCT that is involved in ROS detoxification. We will need to understand if there is a link between CD98HC and KEAP1 and STK11 too, of course. But we didn't see the switch, and it has been shown that KRAS STK11 or LKB1, in fact, promote a switch from adenocarcinoma to squamous. So we know it's not exactly the same mechanism. However, about the ROS, the redox status, we will need to investigate further. We will need, in fact, to test whether XCT will work the same way, and anti-XCT will work the same way.
Thanks, Laetitia. There is a second question from Patrick Brest in the chat. Do you think that the dual function of CD98 may be different dependent on the P53 context? Basically, one having a more preponderant activity depending on the P53 mutation.
Yes. I have to admit that I don't know for sure, but I think that when we have a P53 mutation, and it's a silenced mutation, when we have this kind of mutation, my gut feeling is that it's much more about integrin related and ECM remodeling than when we don't have this P53 mutation. So the integrin part, the modulation of CD98HC will be really important with the integrin relationship, I think. But I don't know for sure because I didn't, I will need to investigate further, but the previous speaker already did show that collagen infiltration is really important for PD-L1 response, resistance.
Thanks, Laetitia. Thank you very much. Are there any other questions from the audience? If there are not, then I will take the opportunity to thank again all the speakers for a really phenomenal session. Humam, Don, Valerie, and now Laetitia, thank you very much for wonderful talks. Also, you make every session chair's heart happy because we stayed well in time today. And I would like to hand over now, I think, to the last part of this session, which is concluding remarks by George Kalin, Mani Sendurai, and Paul Hoffman.